The purpose of the study was to gain a great understanding of the causes of developmental dyslexia. Developmental dyslexia is a common learning disability in which children have difficulty in acquiring reading skills, and they typically struggle with difficulties in single-word reading accuracy and also in spelling, and these are somewhat surprising given their other cognitive skills. The most prevalent theory about dyslexia is that it's a language-based learning difference, and that children have difficulty in learning to read because they lack an understanding of the phoneme structure of the language, which is necessary in order to understand how words are mapped onto their written counterpart. In addition to their phonological difficulties and their reading problems, there are other differences in children and adults with dyslexia that set them apart from typical readers. And these behavioral manifestations have been quite diverse, and one that's been of great focus is the visual system deficit. And our study pertains to the visual system, but particularly the magnocellular visual system. We conduct fMRI studies to allow us to image the part of the brain that responds to visual motion stimuli, because that part is the cortical extension of what people broadly think of as one of two visual streams in the brain that begins as early as at the retinal ganglion cells and carries through the thalamus and then up into cortical areas, particularly an area called V5MT, which is the focus of our study. What we did is our participants, first of all, would lie inside the scanner and do a task in which they were asked to tell us the direction of dots, either to the left or to the right side of the screen. And this elicits activity in area V5MT, and we use that and compare it to a stationary control condition. What we find is in typical readers, children and adults combined, there's a relationship between a person's activity inside the brain in response to visual motion and their reading ability as measured on standardized tests of reading. That in itself is an interesting finding, but it doesn't really speak to the causality of it, and that's what we tried to get at in this study. And so we recruited children with dyslexia. These are children who are at a school nearby here in Maryland at the Genesee School. First time we compared the dyslexic children to children who didn't have dyslexia and replicated a finding of a difference in area V5MT activity as they were doing this visual motion task. This is consistent with previous findings, but it's the next step that's really novel to our study, which is where we begin to try to address the issue of causality.
The first step here was to compare the children with dyslexia with a group of younger controls who were matched for reading level. If the deficit exists in comparison with both the age match and reading level matched controls, it provides very strong evidence that this deficit is causal to the disorder. When we compared activity in V5MT between the children with dyslexia and the younger reading level matched controls, we found no difference in activity, providing the first evidence that this deficit may not be causal to the reading problems. In fact, it's possible that it's a side effect of dyslexia that exists with other deficits, such as weaknesses in phonological awareness, or that it's related to a person's reading experience. To test this, we looked for a change in V5MT activation in children with dyslexia following an intervention. The reading intervention was administered by experienced clinicians for 8 weeks and focuses on promoting awareness of phonology and orthography in order to bring about an improvement in reading. The children also completed a second 8-week period as a control to make sure that any observed gains would be specifically related to the reading intervention. Following the intervention, as expected, the children made gains in reading during the reading intervention period, but not during the control period. In addition, when we measured V5MT activity following the reading intervention, we observed an increase that again was not observed during the control period. Therefore, we believe that reduced V5MT activity is a consequence of reading disability and not the cause.
We think that this research is informative for practitioners and clinicians in helping us understand what are some of the end product, what are the manifestations in dyslexia, but to be able to distinguish those from the actual cause and the kinds of skills that are important for identification and treatment of dyslexia.